Hello aspiring NEET PG warrior, welcome back to the PYQ series. I am here with another video of PYQ series on Streptococcus pneumoniae. You must be thinking that what is unique about this PYQ series, why this PYQ series? So I must tell you the content which I am delivering in this PYQ series is meticulously curated for your PG examination. The contents are taken not only from the microbiology textbook but also from the textbook of medicine such as Harrison, textbook of infectious diseases such as Mandel and Douglas. So rest assured the contents are very precise, comprehensive and tailored to ensure your success in the upcoming PG examination. So welcome back to my YouTube channel Microbiology Simplified. I am your guide and mentor Dr. Amit Kumar Singh. Let's start with the video. Okay, so let's start with the today's topic, Streptococcus pneumoniae, PYQ number 3. So just see the MCQ. A 25-year-old male patient presented with the symptom of meningitis. Microscopic examination of gram stain smear of CSF showed organism as shown in the image below. So which of the following is the characteristic feature of the organism? So to find out the organism, first we see the disease. The disease is meningitis, okay, and they perform the gram staining of CSF, and also the information provided here is the age of the patient. So on the basis of these three information, we will find out the organism and then the correct feature or the characteristic feature of the organism. Okay, so what we can see here in this image, that the organism are mostly arranged either singularly or in pairs. So mostly you can see pairs. Okay, mostly you can see the organism is in pair. Okay, so it is, and the color is purple. So what it is a gram positive, and on the basis of arrangement it is diplococci. Okay, so it is a gram positive diplococci. And also what we can see here, if you just zoom into this the image, you can see the shape of the cocci. It is like this, that one end is pointed, okay, and another end is round, and this round end, this diplococci arranged with this round edge facing to each other. So this will give this cocci a shape of flame. So this is flame shaped, or also we can say lens shape or lanceolate, either flame or lens shape, lanceolate. Okay, so this is a gram positive diplococci arranged in pair and flame shape. So what could be the organism? So first, for that we need to know that what are the common organism causing infection in adults. So if you can go through the previous videos on bacterial meningitis, you can get this list. I'll not go into detail of this list because it is already discussed. So just I will focus in this age group, 2 to 50 years. So in 2 to 50 years, there are two common bacteria which causes infection. First is Streptococcus pneumoniae and second is Neisseria meningitidis. Out of these two, the first one, that is this pneumoniae or pneumococcus, is gram positive cocci, and this Neisseria is gram negative cocci. So obviously, which one is the organism asked in this question? It is Streptococcus pneumoniae. Okay, and also we know that Streptococcus pneumoniae is a gram positive diplococci. So now to answer the characteristic feature of the organism, we need to know. So if you'll see the option, it is about the biochemical properties. Okay, these are all the biochemical properties of the organism. Okay, so to answer this question, we need to know the biochemical properties. So I'll start my discussion with the biochemical properties of Streptococcus pneumoniae. Okay, so Streptococcus pneumoniae is catalase and oxidase both negative. Okay, so this is actually a characteristic of Streptococcus, that all the Streptococcus are catalase negative and all Staphylococcus are catalase positive. So if you see a gram positive cocci, and if it is mentioned it is catalase positive, that means it is staphylococcus, and if it is mentioned as catalase negative, then it means it is streptococcus. Okay, and it is also oxidase negative. A very popular oxidase positive gram negative bacilli is Pseudomonas. Okay, second information here is it ferments sugars, almost all sugars, including inulin. Why I am saying inulin particularly? Because Streptococcus pneumoniae can ferment inulin, but not other streptococci. So this helps differentiating between Streptococcus and pneumococcus. So Streptococcus cannot ferment inulin. So this is a characteristic of pneumococcus now. Also this again the third point, that is optochin. What it is? Optochin sensitive. It is optochin sensitive, that is it means it is sensitive to this antibiotic, which is actually ethyl hydrocuprin. In examination, instead of mentioning optochin, they may mention ethyl hydrocuprin, so that's why I'm mentioning it here. So pneumococcus is optochin sensitive, whereas the other streptococcus are optochin resistant, and also it is bile soluble. Okay, so it is bile soluble. So if you add a suspension of streptococcus pneumoniae into 10% sodium deoxycholate, so it will dissolve, it will become clear. That indicates it is bile soluble. So this again is bile solubility is characteristic feature of Streptococcus pneumoniae. So bile solubility, optochin sensitivity, ferment sugars including inulin, and catalase and oxidase negative. So these are the characteristics. So now let's see that whether we can solve the question or not. So glucose and maltose fermenter. Also, as I said, it ferments almost all the sugars including inulin. So glucose and maltose fermenter, this is correct. And non-inulin fermenter, so this is incorrect. Okay, because it is inulin fermenter. Catalase and oxidase negative, this is correct, but inulin non-fermenter, again this is incorrect. So this option is also not correct. So these two options are not correct. Catalase and oxidase positive, this is incorrect, and glucose and maltose non-fermentant. So this full statement is incorrect. And now the fourth statement, optochin sensitive, correct, catalase negative, correct. So the correct answer is optochin sensitive and catalase negative. Okay, so apart from this, they may mention anything from the disease, from the epidemiology, from the pathogenesis, from the vaccine. So that's why you need to know the other important points about pneumococcus. Okay, as I told you, I will not just discuss the question and its answer. It is now it is done. You may stop the video here itself if you just want to know the answer of this question. But if you want to know about the disease, if you want to know about the organism, so then proceed with the video and you will definitely get the detailed information which may be asked in the examination. Okay, so the key points, so morphology, as I told you, it is gram positive diplococci, and what else it is important to know, it is capsulated. It is capsulated. Okay, it is capsulated, and the most important antigen because of virtue of this capsule is capsular polysaccharide, and this capsular polysaccharide is also known as specific soluble substance. Okay, triple S. So SSS is found in pneumococcus, and on the basis of this capsular polysaccharide, it is classified into about approximately 100 serotypes. Okay, and this serotyping is done by any of these three methods, that is Queuling reaction, lattice agglutination, and PCR based method. So what is Queuling reaction? Examination I have seen question asked on Queuling reaction. The Queuling reaction is nothing, just by adding a suspension of this organism. Okay, that means suspension of Streptococcus pneumoniae plus its homologous antiserum along with loop full of methylene blue. So what will happen when it is mixed with this antiserum? The capsule will apparently swell, and that can be visualized by using this dye methylene blue. So apparent swelling of the capsule is indicated by the methylene blue, and this reaction is called as Queuling reaction. So Queuling reaction is basically to demonstrate the capsule. Okay, and this can also be demonstrated by latest recognition and PCR based methods such as multi-locus sequence typing, multi-locus enzyme electrophoresis, pulse field gel electrophoresis, and amplified fragment length polymorphism. So these are the various other methods by which we can serotype the pneumococcus. Okay, and the source of infection, to your surprise I must tell you, is the respiratory tract. This is not the surprising thing, respiratory tract. So what is surprising thing? That the respiratory tract of carriers are the more important source of infection, and the less often is the patients. Okay, so this is less often. So this is not the most important. The most important uh, is the are the carriers. Okay, and it is transmitted through, obviously, through droplets. Okay, and the serotype associated with disease. So the most virulent is type 3. Type 3 is most virulent, and this type 6, 14, 19F and 23F, it is, these types are associated with invasive infections. Okay, and this type 1 to 8, this type 1 to 8 is associated with around 75% of pneumococcal pneumonia and 75% pneumococcal pneumonia and tracheobronchitis. Okay, and this type 6, 9V, 14, 19 and 23, this is frequent cause of meningitis among children. So they may ask in the examination that which strain of pneumococcus is associated with meningitis, with invasive infection. So if you see, the strains which are associated with invasive infection are also associated with meningitis in children. Okay, and then pneumococcal pneumonia and tracheobronchitis is commonly associated with type 1 to 8, and the most virulent among them is type 3. So these may be the options. So that's why I told you the options may change in the future examinations. So you just don't memorize the question and its options. So option may change, so better to go to the topic. Okay, now the pathogenesis. So if you see, firstly it will, as I said, that the respiratory tract of carrier is the source of infection and through the transmission through droplets. So it will initially, it will colonize the nasopharynx. Okay, and then it will survive there because of bacterial capsule and by formation of biofilm, and then it spreads. It may spread via blood stream to distant sites such as brain, joints, bones and peritoneal cavity, and it may spread locally to the mucosal surface causing otitis media and pneumonia. And it may spread directly to CNS, but that is very rare. So direct spread usually occur through fracture of the skull base. Okay, fracture of a skull base, and the more common mode of transmission of infection to meninges is hematogenous spread. Okay, so remember, this pneumococcal meningitis is usually secondary to infection, such as the primary infection such as pneumonia or otitis media, because from these sites it will spread hematogenously to reach to the meninges causing meningitis. Okay, so remember, this meningitis is usually secondary. So what are the diseases which is caused by pneumonia? Lobar pneumonia, this is most common. And in acute phase of lobar pneumonia, you can see rusty sputum. What we can find, a rusty sputum. And this rusty sputum, apart from pneumococcal pneumonia, lobar pneumonia, is also a characteristic feature of a parasitic infection, which is a lung fluke. And what is that lung fluke is? Paragonimus. Okay, So in case of paragonomous infection, we can also find rusty sputum. So rusty sputum is DD of both pneumococcal pneumonia and paragonomous infection. Okay, an early stage we may see bacteremia, and later on toxemia may occur due to diffusion of what? Capsular polysaccharide. Okay, and the most common focal complication seen in lobar pneumonia is acute empyema. Okay, so most common focal complication of lobar pneumonia, acute empyema. Okay, and bronchopneumonia, it is almost always secondary. Remember this. Bronchopneumonia is almost always secondary to a primary infection somewhere else. Okay, and in aged and debilitated patients, it is the most frequent terminal event caused by pneumococcus. Okay, and it is commonly associated with the acute exacerbation of chronic bronchitis and tracheobronchitis. Okay, so it is commonly associated with acute exacerbation of COPD like chronic bronchitis or tracheobronchitis, and the similar thing is seen, I told you in the previous, I think in the PYQ number 2, or no, in PYQ number 1, it is seen by which bacteria? Haemophilus influenzae. Okay, so remember this, acute exacerbation of COPD is seen in pneumococcal infection and Haemophilus influenza infection. Okay, remember this. And then it causes meningitis, and as I told you, it is usually secondary, but primary may occur in case of skull base fracture. Bacteremia is also present in case of meningitis and certain other suppurative lesions. These otitis media and sinusitis due to local spread, and this pericarditis, suppurative arthritis and peritonitis due to spread through blood stream. And it is also known to cause keratitis, ocular infections such as keratitis, dacryocystitis, conjunctivitis, endophthalmitis may also be seen in infection caused by pneumococcus. Okay. So how to diagnose? Microscopy, as I told you will find gram positive diplococci arranged in pairs. That diplococci, that means arranged in pairs and flame shape. Okay, what else? Culture, we can isolate the organism. So culture should be done on which medium? It should be done on blood agar. As it is fastidious organism, it should be done on blood agar under 5 to 10% carbon dioxide. Okay, and the colonies, if you know that streptococcus are classified on the basis of hemolysis, so this pneumococcus will show what type of hemolysis? The colony will show alpha hemolysis. Apart from streptococcus, which type of pneumococcus, which type of streptococcus will show alpha hemolysis? Viridans group. Okay, and initially the colonies are dome shaped. Okay, initially the colonies are dome shaped, but on longer incubation, what we can see, we can see this dome shape, like this dome shape will become flat and with raised edges. Okay, the edges will be raised and with central umbonation, that is centrally it will raise like this. So it will give it an appearance which is called as carom coin appearance or Draughtsman colony. Okay, so this carom coin appearance will not occur initially, but it will occur on longer incubation. Okay, remember this. And then we can also demonstrate the capsular polysaccharide in CSF, in blood, and in urine too. Okay, so remember this, in urine also we can demonstrate the capsular polysaccharide. CRP is an important test which can be done in case of Streptococcus pneumoniae. Apart from CRP, an important marker of invasive infection is procalcitonin. And what is the use of procalcitonin? Procalcitonin can be used to monitor the progress of treatment and prognosis. So it is an important marker because it will also tell that whether the treatment is going on correctly or not, and also how the patient responding. Okay, so procalcitonin become an important marker, especially in invasive infections such as meningitis. Okay, the vaccines which are available for pneumococcus, the first is capsular polysaccharide vaccine. It is also known as PPSV. What is that? Pneumococcal polysaccharide vaccine 23. Why 23? Because it contains 23 serotypes or 23 valents, okay, of pneumococcus, and it is recommended especially in United States for all persons of age more than 65 years. Okay, and also it is recommended for the persons of age group between 2 to 64 years of age with underlying conditions such as absent or dysfunctional spleen, sickle cell disease, celiac disease, chronic renal, lung, heart and liver diseases, diabetes mellitus and immunodeficiencies like HIV. So these are the conditions in which it may be recommended to persons below 64 year of age but above 2 years. So it is not recommended for children below 2 years. So it is not recommended for children below 2 years. Okay. So what is for below 2 years children which is used in vaccination in India is the polysaccharide protein conjugate vaccine, also known as PCV, pneumococcal conjugate vaccine, polysaccharide protein conjugate vaccine. So initially a 7-valent PCV was introduced in around 2000, and then later on 10 and 13 serotypes PCV was introduced, and these serotypes include the strains which causes invasive pneumococcal disease and antibiotic resistance among young children. Okay, so these vaccines will include those serotypes which causes invasive infections and antibiotic resistance among, especially among young children, young patients. Okay, in US this PCV 13 is followed by PPSV 23, that is the first polysaccharide vaccine for all immunocompromised adults and children. And again, there are two new extended valency vaccines are available. The first one is contain 15 serotypes and the another one contain 20 serotypes. The first one is Vaxneuvance and second one is Prevnar. So these two are also available right now. Okay. So this is about the Streptococcus pneumoniae, and I believe that most of the question will be within this content only. And in examination, definitely if anything asked on pneumococcus, we will be able to answer if you will go through this video from start to end. Okay, so don't miss anything in this video. Make a notes of it the best way. Just go through the video, make your own notes and summarize it, and then revise the notes. Okay, you may go through the video again and again if you have any doubt, but I want you to revise through your notes only. Okay, so all the best. Just prepare with me. Take care and wait for the next video which will come tomorrow.